This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 690, recorded on December 4th, 2020. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello, Vincent, and everybody else, too. The weather outside looks pretty grotty. I haven't been outside to actually see what it's like, but it feels cold just by looking at it. There's no rain, but it's just one of those days that I'm glad to be inside podcasting, I'll tell you that.
And so are we. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. 55 degrees and sunny, headed for 57. That's today's high, which for around here is pretty chilly, but it's okay. Sunshine, I'm happy. It's good.
From Western Massachusetts, Alan Dove.
Where we have the same weather as Austin, Texas, apparently. It's 56 Fahrenheit, 13 C, and it's partly sunny, but it's supposed to cloud over pretty soon tonight and then rain and then turn over to snow, and we're supposed to get 3 or 4 inches of snow.
Wow. Cool.
Yeah.
Gotta get your snovel out. What is it called? Wovle?
The wovle? Yeah.
Wovle.
I actually have an electric snowblower, but when that breaks, I've got the wovle as a backup.
I will bet you the Wovle arc is pre-pandemic.
Oh, it is.
So There are a whole bunch of listeners. There are 90% of our listeners who don't know what a Wovle is.
Or at least have not heard about it on this show. This is something that has been
If you haven't heard about it on this show, chances are you don't know what it is.
Yeah.
Also joining us, our guest today, I don't know how many times you have been here, Amy Rosenfeld. Welcome back.
Thank you for having me. I've been here every month since October.
Is that right?
Yeah, I've been here once a month, every month.
You were here before a couple of times, right?
I was here 3 times beforehand.
Okay.
But
And Amy has taken time from her experiments to help discuss papers today.
And so are we going to keep calling Amy a guest, or are we just going to, at some point, start referring to her as a rotating host? I mean,
I think she's family.
She's a
actually.
regular.
I think
Could
she's
be a
family.
rotating host if you'd like.
yeah, that sounds good. I
Yes.
don't like guests. It makes you sound like you should all get out the fancy dishes and stuff. That we need to get into the dining room, kind of a kitchen kind of gal.
So we have also another individual on this podcast. We have a cat.
Yes, that is Suki in the background, who is currently the target of a cateriaphage infection, as you can see. So people have previously seen Oliver on the show. This is Suki, who is the mellow, aloof cat. So she's just impersonating a seat cushion back there.
All right, we have one piece of news which Amy sent me yesterday. So it's an article in New York Business Journal, which I have not read. It's giving me offers to sign up, of course. New York City will create Pandemic Response Institute to prepare for future outbreaks. Do you know anything about this?
I read about it yesterday. Like, I was on the train going to the wine store and it came across, and I thought it was kind of a cool idea, but it's very rudimentary at this particular point in time. So
They have no names of people that they want to add to this group?
no, but it's not exactly clear. They're going to have a diagnostic arm and then some economics and some other stuff for detection and management and stuff.
Here's what they're going to do. They will focus on building technology to detect, track, and monitor health issues, research diagnostics, therapeutics, and outbreak modeling, training public health business and community leaders in pandemic prevention and management, and creating community-based partnership models to build local-level infrastructure, health infrastructure, improve emergency preparedness, and improve health outcomes.
So I think it's a great start, but I think that they need to have a 5th component to it, which is how to deal with the economic consequences of shutting down businesses and also schools. So maybe it's a 6th component.
Do you want to be the director?
No, I think this should be, Ian wanted to be the CDC so he could practice here.
And they're going to put out proposals for partnerships with institutions. I assume they mean academic institutions. So depending on how much money is on the line, let the jockeying begin.
Yeah, but I think it's like a great start and really creative to look that we never, ever experience this again. And I think that learning from this and not forgetting it, which is what we tend to do, is a good model for other places, to take in, because you don't want to ever be in this position again.
This is also, it seems like an appropriate thing for New York City in particular to do, given the city's history with robust public health programs. I mean, they've typically been, not always, but frequently they've been at the forefront of stuff like this, and so it's good that they're interested in keeping that edge.
As a transportation hub, they were also ground zero for this pandemic, and that would likely be the case for future pandemics. So it's appropriate.
Yes.
It's kind of interesting to note also that since Lassa fever was first introduced to this country at our medical center without further consequences, nothing more was heard about that except from the national level to establish peace facilities, P2, P3 facilities. New York City didn't click to the fact that this thing ever got loose in the general population. All kinds of chaos would have erupted. So we're lucky that didn't happen. But that was one chance that they had early on to establish what Amy just talked about.
Well, I think actually the HIV epidemic might be a better example with the New York City Gay Men's Health Network and everything. They really mobilized to go out into the gay communities, to educate people in the bathhouses and stuff. So I think that is actually maybe more a direct application of like the power that the citizens of New York City could have.
So the thing I hope they avoid is a duplication of efforts with what will hopefully soon be a functional CDC. I mean, I'm sure part of the motivation behind this is that for this pandemic, the CDC was mostly out of play for political reasons. And so New York City had to take charge of it in a way that they hadn't previously really, and on a scale that they hadn't previously dealt with these issues. But I hope this won't become just a duplication of efforts of stuff that's already being done at CDC. And then 10 years down the road, somebody says, well, this is a duplication of efforts, so let's cut this whole thing. But I think there is a legitimate niche for this sort of thing that could be tailored specifically to New York's interests, and that could become a real center for these types of studies.
And I'd like to see this coordinated with lots of national and international health agencies as well, because I think if you don't, then we will be the recipient of all kinds of inputs, which we won't know what to do with because there's nowhere to feed back to that source, which I think was the case with China, for instance, and other examples as well, by the way.
So I have two questions. First, is this going to be something that large cities in the U.S. do? Because you can imagine Chicago, Atlanta, San Francisco, Boston might all want to have something, right? Philadelphia.
Miami.
Seattle.
Yeah.
Not Austin, right?
No. We're too busy playing music.
Okay, so that might be a good idea. The other is, who's going to pay for this? There's no mention of money, right?
No, he hasn't talked about how he's going to fund it. But you can imagine that it could be funded partially out of your state taxes. It could be funded partially by like donors like the Gates Foundation and stuff. So I don't know that I agree with Alan about it overshadowing the CDC, because I don't think that the Gay Men's Health Network in the 80s for HIV overshadowed the CDC. And I don't think that we walked away feeling that the CDC was as broken back then as we feel it is today.
Right. Okay.
So I think that they can work in parallel and synergistic. And this gives you a local contact point versus a national contact point that you can imagine all coalesces into the national network and then national decision of how to address
I would agree with
pandemic.
that. I just I hope that the people organizing this are aware of that history and structure this in a similar manner is my concern, and don't just retread what could be delegated to the CDC.
Yeah.
But a lot will also depend on the funding sources.
Well, the other thing is one has to recognize that Blasio is a two-term mayor. He's done in 2023, I think, or 22. So,
I hear Rudy Giuliani is going to run for mayor.
not again. Not after
If he's a free man,
pardoned.
yeah.
Says de Blasio could invoke the Bloomberg rule, but that's not going to happen.
No.
No, he's not Bloomberg. No. So I think it's a great idea. I think it's a good thing to have a discussion, but whether or not it gets implemented under him, I highly doubt. But I do think that the next mayor should take this and bloom it into something because, the city was disproportionately hit, and the neighborhoods like up here where I live is kind of sad sacky.
Yeah. And a way this could be set up that would reduce the funding requirements and also the risk of redundancy would be as more of a virtual institute. I mean, maybe have a small office that coordinates it, but really work with the institutions like Columbia and Weill Cornell and NYU and such and Mount Sinai that are already there, that often have people who are doing these things, and have them coalesce around this, meet periodically, and then when you need them, you can call them in, kind of volunteer fire department approach.
It also shouldn't compete with the city health department. I think it should probably be below the city health department in some agency that reports back, because you've already got the network set up for healthcare workers and door-to-door surveys and things of this sort. School nurses, all those things are already in place. Why duplicate that part? This is, just in case something bad happens.
Well, and you don't want to see it becoming a zero-sum game with the budget. Let's pull money out of the health department and put into this.
Exactly.
Don't do that.
That's exactly right.
All right, thanks for bringing that up. Anything else we should know about in the news going on?
Yes, Tony Fauci has risen to the level of the new administration's health advisory board. And that's a great thing to see, because I knew that Biden would move in the right direction, but this is overt evidence in that direction. Pleased to see it.
All right. We have two papers for you today, and they're both on, guess what?
Polio?
Smallpox.
Plant viruses. We're doing plant viruses, right?
Racaniello.
Rich, we have a smallpox paper coming up.
Is this the new thing from Clarissa?
I don't know, but the CEO of Tonix Pharmaceuticals sent it to me, and he said this nails down where Jenner got his virus from.
I think this is the one. I just saw a post today from Clarissa. So, we can share this, and that can go in. Like I said, I've got a whole smallpox show ready to go, and I can add it to that folder.
So he actually will join us. He's the CEO of
Tonix,
the company that made the horsepox DNA, infectious DNA. So,
or had Dave Evans do it. They collaborated. Yeah, Evans did it.
So do we now know for sure that vaccinia came from horsepox?
That seems to. I need to read this, but in the abstract, that seemed to be the bottom line. Basically,
we'll have a show on it.
Or there's horsepox in there,
right?
Somewhere.
Right.
Okay.
All right. First paper was sent to us by Tony Schounts, who was on TWiV, I don't know, some time ago. I won't read his letter until we go over the paper, because it gives away the whole punchline. This is published in PNAS. Broad host range of SARS-CoV-2 predicted by comparative and structural analysis of ACE2 invertebrates. A lot of authors from a lot of places. The papers are having more and more authors, it seems. which is great. It's a collaboration. But for TWiV, it's hard because my first author is Joanna Damas. The last author is Harris Lewin. And it's from all over the place.
And we're not trying to shortchange all the people in between. We just
there's just too many. Well, not too many for me. Anyway, so the point here is we need to know what other animals can be potential reservoirs, right, of SARS-CoV-2, because right now we know a handful. And they try and do that here in a way that does not use any liquids.
Not a single wet lab experiment.
Well,
crushes my heart.
I mean, there may have been beer.
Yes, that's true. And wine or something, yes, or water. But this is not to disparage computational biology, as Eugene Koonin would say. But there's a limitation, as you will see here. But they say if we look at ACE2 and spike, and we take spike from SARS-CoV-2, which interacts with ACE2, and if we look at the different ACE2s, now all these animals they look at, the ACE2 has been, the ACE2 ortholog, the gene sequence has been made, so you can predict what the protein looks like. And can we say whether this would work or not, whether spike would bind to them? That's the bottom line. All right, so it's predictive. And they have 410 vertebrate species that have ACE2 orthologs. We got mammals. This is going to test our zoology. Birds, fishes. And no, that's the time when you can say fishes.
Yes.
I like that. You don't always have to say fish. I'm eating multiple fish tonight for dinner, but we're going to classify different fishes. Right, Dixon?
Right.
Get to unmute himself.
I know, but he was so far away from the computer.
It's chilly.
He had to commute to the microphone. For those who are not watching on video, he had to kind of lean forward to the microphone, then unmute himself, and then say right.
He's chilly.
He woke me up. What can I say?
Fishes.
He had to call Dixon in from the outfield and have him do that.
And reptiles and amphibians. All right, so they all have ACE2 orthologs. Sequences are known. And then they look at 25 amino acids, which are known amino acids in spike that bind ACE2. And, they take 25 amino acids from human ACE2 that are known to interact with spike of SARS-CoV-2. And they say, are they similar in these different animals? And they score it. They get a set of rules, and they have 5 score categories: very high, high, medium, low, and very low for the possibility that these would interact, that spike would interact with ACE2 of these animals. Okay, and that's really the bottom line. And they have a couple of tables where they have, very high. Guess who's at the top of the list? Of course, Homo sapiens. Gorillas, gibbons, chimps, bonobos, mandrills, sooty mangabees, etc. And then high, this is based on the number of residues that are similar out of those 25. Lemurs, deer is in the high, gray whales, porpoises, narwhals, one of my favorite animals.
Yeah.
I love narwhals. Muskrats, killer whales. Wow. Okay, so that's high. Then we have medium, more lemurs, marmots, golden hamsters, which happen, I think hamsters are a really good model for SARS-CoV-2 infection. I don't know if it's the golden hamster.
It's the Syrian golden hamster.
Is that the same one as this?
I think so, yes. But so far you've not said a bat.
Where's the bat? I haven't said a bat yet, no.
No.
A sperm whale, which is another one of my favorite organisms.
There she blows.
Yaks. Okay, so then we move into medium. Oh, that's still medium. Goats, sheep. If anyone has a favorite animal, tigers, leopards, cheetah.
One of the medium risk animals has a common name of Nancy Ma's night monkey.
That's good.
Which it sounds like the title of a movie or something.
Then we have howlers.
Then we have deer mice.
Deer mice.
They're, yeah.
Where are they?
Right here is the.
So they're
Peromyscus.
The little pocket.
Peromyscus?
Yeah, the little pocket mouse and then the deer mouse.
Peromyscus maniculatus. Yeah, those are the ones that Tony Schounts says are really infectable. So they're not medium, I would say. I say they're
high.
Pretty high, which immediately tells you that this is not, perfect, right? And then we have low, rhinoceros, camels, pandas, polar bears, chinchillas, horses, whales, more whales, dogs, elephants. Yeah, there are a whole bunch here. I'm just giving you an idea. Then we have very low, sea otters, sea lions, European mink.
Chinese pangolin.
Chinese pangolins.
Now, one of the things that stands out in the very low list is all these bats.
Yes, there are a lot of bats.
That's what I said.
There are a lot of bats here.
I pointed that out.
Yeah, well, here they are, fruit bats and all kinds, yes. But,
no, but even
so
here.
Amy said you hadn't said bats, and I just wanted to point out it wasn't because he'd missed it in the list. It's because a lot of them ended up in the very low risk category.
Yeah, there are a lot of, so all the bats are in the low risk, right, category? Yeah. Big brown bat. But the one that we really want to point out is almost at the bottom of the very low list, which is Neovision vision. American mink. And we know that, and in fact, this is the mink that's farmed in the Netherlands and Denmark and Minnesota, or not Minnesota.
Or perhaps we should say used to be farmed in the Netherlands and Denmark, right?
Yeah. And
so this is the best characterized spillback.
Yes.
To date.
Now, I should point out that in the little significance box after the introduction, the final sentence says, given the limited infectivity data for the species studies, we urge caution not to overinterpret the predictions of the present study. So they're quite aware that this is a computational theoretical type of paper.
And I think the mink and others point out that the limit is severely limited. And so I asked Amy this morning, why is this in apparently a high-profile journal, Proceedings of the National Academy of Science? And what was your answer?
Well, I had two.
You had two answers?
I had two.
What were they?
So one in SARS-CoV-2, and two, people get seduced by the pretty picture and the idea of computational biology.
I have a third one. It's in the fine print on page 1 where it says this article is a PNAS direct submission.
That is true too.
That's right.
But I
so
did
there's for people who don't follow the literature, Proceedings of the National Academy of Sciences is a very prestigious journal, but it has a back door installed that's different from a lot of other journals, which is that a member of the National Academy of Sciences can just put a paper in it.
It's true.
So it doesn't go through the normal peer review stuff. You can just publish it, and that doesn't mean it's a bad paper, but it means that it may have not cleared the same bars that it would have had to clear to end up in other journals.
Is it not true that currently even direct submissions, the submitter must get a couple of reviews?
There is a procedure.
There is a procedure.
The point is that it lowers the...
it's not the same procedure.
It's a bit of a shortcut.
Yeah.
Do you think neuropilin might also play a role in this? And that if that was included in the survey, perhaps some of these would have risen to a higher level?
Well, actually, that's the other main point, is that this and many other papers assume that if you have ACE2, that's it. And
that's the whole story.
All you need is ACE2.
Right.
But
I remember raising neuropilin as an issue a long time ago in our show.
Well, neuropilin may be a entry cofactor, but it's all entry. But what I'm saying is, a tenet of virology is that you absolutely need a receptor to get in cells, but even then it's not enough because the internal environment of the cell has to be permissive. And they do not address this. They just say in the discussion, well, other things might influence reproduction like the immune response. Really, just the immune response. That's
like the biology of the animal.
So I find that this is a problem because these people don't understand this, and they're doing blind studies, and they have no idea about the biology of the virus. Okay, and so having a receptor, look, poliovirus receptor is on many, many different tissues, and the virus doesn't reproduce in them. There's something else that's going on. And that's the case for many viruses. But I've noticed in this pandemic, everybody publishes on ACE2 being high or low or here or not, and that's the only story. And that's a big problem as far as I can see.
But it's not this pandemic. It happened during Zika. It happens during EV68.
Yeah,
it happens during everything. But like
it happened during HIV.
Right. But like even a better example than polio in humans.
There can be no better example.
Oh, sorry. The God has spoken. No, but for years we've tried to make a rhinovirus model using mice. And you can put the rhinovirus receptor on mice and it gets in, but it doesn't reproduce. It actually has to be adapted. And some of the adaptations are in the non-structural proteins that make the virus factories for replication more efficient. Wouldn't you say that was what Julie found? So
it's one of my former students, right?
Yeah. So this idea that receptor is the solely importance is not correct. And none of the studies actually that they cite in here actually take the RNA and transfect it. And we did that actually for our Zika work. We saw one isolate that didn't replicate, and we wanted to know, was it because it used a different receptor or because the internal environment wasn't permissive for the assembly of the particle? So David and I electroporated RNA that we isolated from the virion into our organotypic brain slice cultures, found that we saw one cycle of replication, and then it stopped because it either needed a different receptor or it needed some factor to disassemble the particle before translation. So
I would go ahead.
This is just annoying as a virologist, actually. It's insulting.
I would like to color a little bit the statement that you need a receptor for the virus to get in by saying that in some cases, and I'm thinking, of course, of my favorite vaccinia virus, really all you need is some kind of surface binding. In the case of vaccinia, there's like about 4 different ways you can bind, and they're all to pretty vanilla substituents on the surface of the cell. So as long as the virus can kind of stick there, then things happen, okay? It doesn't have to be necessarily this really highly specific, unique receptor sort of interaction.
Yeah, some viruses do have very specific receptors and others just need to stick.
And there was a couple of other things about this. I probably didn't read it closely enough, but it was difficult for me to tease out of this whether they took into consideration really broader structural considerations on the interaction. The bulk of the data are just that here's these 25 amino acids that we know make contact. And is there, what is the similarity or differences in the ACE2 receptors of all of these other things, and then draw conclusions from that. But really, the virus receptor interaction is more subtle than that. And as I recall, in particular, the whole time I was reading this, I remembered our discussions with Ralph about actually the discussions surrounding whether or not SARS-CoV-2 might have been engineered in the laboratory. And one of his comments was that based on his understanding of the SARS-1 receptor interaction, and the contacts made there, he never would have predicted, the interactions for the SARS-CoV-2 receptor or spike protein and ACE2, that involved a whole different spectrum of amino acids and subtle differences in the structure. And as far as I can see, this doesn't really address those sorts of subtleties. And that may account for some of the differences. I mean, it'd be really interesting to see, in fact, what structurally, at a structural level, what the interaction is with the mink ACE2, and see why it is that there are these differences.
Yeah, well, even to take Ralph, the remembrance of what Ralph said one step further. Like without that knowledge of the whole 3-dimensional interface and structure of the contacts and shape of the molecules, Ralph would not have been able to computationally predict which 2 amino acids to change in order to mouse adapt the virus. So he used all that information initially to make his mouse-adapted virus, and then he passaged his mouse-adapted virus to get it to be even more mouse-adapted. But those first two amino acid changes, which are almost next to each other, they're like 499 and 501 in spike. Those changes he had to use, he didn't just throw up a dart. He used all the information to do it.
Yeah, for sure.
So you can't, this paper is under appreciating the intricacies and delicacies of receptor and virus interactions.
Absolutely well put, yes. Underappreciating the delicacies. Is that what you said?
Yeah.
The other thing I would have done here, for many of these species, I'm sure cell lines are available, right? You could just take them and infect and say, now that requires a BSL-3, so it's not so trivial, but you could
use the pseudotyped virus.
Probably pseudotype.
Oh, you could do pseudotypes, that's right. So it wouldn't be so hard to complement this and say, is this prediction in line or not, right? Because if you took mink cells, you would find, oh, they are very highly susceptible. And so, you know, computational biology has its place, and it's especially in phylogenetics and in evolution. But to make predictions about biology, you really need to complement them with some experiments, I think.
Yeah, but not a single person.
In the author's defense, if I may, because we've spent the past 15 minutes kind of harshing on them, well, with some very appropriate gripes, I think. But this is not a bad study, and it's not a bad idea to look at this. And certainly, if you're locked out of your lab because of the pandemic, and all you've got is sequence information, and what you do is bioinformatics, this is a great place to start. But I think what's coming out here is we're kind of being reviewer number 3. And saying, you know, all right, this is a nice preprint you've put up. What we'd like to see for a publication is this, and this, and these experiments, and you need to tone this part down and account for this. And I think you could really potentially go somewhere with this by finding, especially if you did that what Vincent just suggested, where you do an experiment in cell lines and maybe find out, oh, you know, we scored bats really low, but it turns out we can infect bat cells. And so we need to tweak our model a little bit. And you might even be able to go back to the computational side and build better predictive algorithms out of that, which could be a really productive line of investigation. So this is a fine starting point, but it probably shouldn't have been published as a paper in a peer-reviewed journal just yet.
Well, I want to be reviewer number 4.
Number 4.
So this, to me, is one figure of a paper. That's it. One figure. And I can explain to you why I feel that way. So one, during Zika, a lab at UCSF wrote an entire receptor paper in Cell Host and Microbe about, AXL being the receptor, because it's on the tissue and it's in high expressing regions, blah, blah, blah. And that screwed up the field tremendously for many years. And even today, we're not clear what the role of AXL is in Zika biology. And there's a lot of data that says when you get rid of it, doesn't prevent virus from infecting the cells, perfectly fine. So it took a long time, and it's inhibitory to the development and the progression of the field. And especially during a pandemic, when you want to move as fast as possible, you don't want to be burdened by weeding out these papers. And they do address the bat situation. So they say that these cell lines from big brown bats and Lander horseshoe bats and Dayton bats could not be infected by SARS. Okay, so what did you do here? You put virus on, you incubated, you washed it off, and then you did a plaque assay? Or what exactly did you do?
That's a reference.
It's a reference, right. So when I went back and looked at those papers, they did like some kind of RT-PCR usually to look at virus RNA. That's not exactly the full picture. As I said, you had to transfect in the RNA and define the cell line.
It's true. Yeah, because...
Which is why David and I spent all of that time doing those hard experiments for Zika. We didn't want to make a mistake. We didn't want to impede the progress of the field.
So what you're saying here is that they take these bat cell lines, they don't they're not infected, but that could be because the receptor doesn't match or there's an internal block, right?
Right.
And they have to sort that out. Yeah, I agree.
Right.
I mean, I think the bottom line here is that
And not a single one of these authors is a virologist. They're from the Genome Institute. They're from the Cancer Institute.
Yeah,
I'm glad you know how to program your computer. You're much better at it than I am. But you're probably not as good a virologist as the five of us.
even Dixon.
Dixon, are you commuting or what?
I want to say one more thing. As if you looked at this and you were a mink farmer and you saw a mink way down, and you go, no problem, right?
Right, no risk.
That could be disastrous. So I think you have to be very careful with this stuff. I want to, now I can read Tony Schountz's original email. He sent it to me. He said, this is a predictive modeling study based on the supposed known binding residues, which he has in quotes. They scored American mink is very low. These are the mink that are used on mink farms in North America and Europe. And it's just another example of, what's this?
Wag.
Wild-ass guess.
Wild-ass guess.
Oh, I didn't even know that. That
So
a lot of the
it's called
modeling.
the Wagner method for wild-ass guess, no
Yeah.
explanation
Right.
required.
We've talked about swag before, which is a subset. It's a scientific wild-ass guess.
Another example of a wag that a lot of the modeling papers do. In the end, the real virology has to be done to really know species susceptibility. Amen. Okay. You know, we're virologists, folks. We like to see the things done properly.
Well, it leads to huge.
So then you may
basic
ask,
problems.
why did you talk about this? Because we want to illustrate the pitfalls, the potential pitfalls. We don't want to just show you all the beautiful science. We want to show you the warts. Well, this is not a wart, but
No. And as I said, I do think there's something good here. It just needs more. And as Amy said, this is one figure of a paper. So this is a nice start, but you can't just run with that.
Right. And when it does is it pollutes the literature. So it makes my job much harder.
Yes.
No, it's not my
No,
job particularly, but
Yeah.
as a virologist or a member of the scientific community, it makes it much harder. Because when I write my paper and that it does all these experiments, I am then going to have to argue why my paper needs to be published because this will be first, and we don't like to publish duplicate things. And then if I want to go into a grant and write a grant, the study section is going to send back and say to me, well, you didn't address this. Why, and then you have to sit down and you have to spend a lot of time contemplating how to address it and not sound like a curmudgeon and belligerent about yo, let's get real here.
what was that word? Yo?
Yo. Yeah, because,
Is that a New York thing?
yes.
Yes.
Okay, let's move on. But
Actually, it's more Philadelphia.
really?
Is it?
It's from Rocky. Yo.
Yo.
Really?
I've certainly heard it plenty in New York.
Never heard you
I
say
think I
yo
heard it plenty
before.
in Philly, too.
Well, I was
So
trying to be
it's
polite
Steve's thing.
this time. Usually I'm not.
Borrowed.
Pretty well.
Borrowed
I
from
would not
Sylvester
be that
Stallone.
I have to say, though, so this argument I hear a lot with respect to Amy's, the virus we work on, She always says, this is going to pollute the field, make it harder for us to write papers and grants, and it's not right. And that's, once something is published, people tend to think it's right, and it may not be. So
Yeah, I've dealt with that kind of thing before. There have been a couple of these incidents in my field where, in fact, with respect to a couple of issues, you get laboratories or authors who have their pet thing that they're trying to use to make a name for themselves that's just dead wrong. Okay. And I found myself in the position sometimes of being sort of the go-to reviewer to keep this stuff out of the literature. Okay. You become a real expert.
Trust science, don't trust scientists.
Yeah. Because
you go.
it can be a problem.
Well, it's also a problem in the fact that, I want grants and I want papers. But for instance, my virus, it's horrible to see a 4-year-old get infected and become a quadriplegic who needs a trach and a breathing tube and a feeding tube and is going to be in a wheelchair for the next 80 years because my ego needed another paper to get my R01 or something. And then I published some ridiculous crap that is not biologically significant. And what's the word I'm looking for? Extrapolated from tissue culture cells, some ridiculous process or property of the virus. My goal is not yes, I would like papers and grants, but my goal is to push the field forward so that and eventually we can help these people who have the disease. And I think we've lost sight of that.
Really?
Yeah, the goal is truth.
You don't want to make a huge lab and get famous and have a lot of money?
No.
Just kidding.
No, that's not my goal. My goal, I'm lucky that I'm really curious and I like science and I love to do experiments. But the end of the experiments that I want to do is to answer a question to help understand why, some disease occurs or some horrific phenotype occurs. Like, you get all these strokes because you have all these mini thrombotic events. Like, why does that occur? Not.
Speaking of mini thrombotic events, let's go to our next paper.
Yeah.
This is from Neuroscience. I debated doing this on...
Nature Neuroscience.
Yeah, Nature Neuro. I debated doing this on Twin, but more fun if we do it because we can learn something. Because this is...
it seems like it's more of a virus paper than a
really is.
neuroscience paper.
But on Twin, I need to find...
In terms of learning something, I have to say, having struggled with this, I have a lot to learn.
Yeah, well, we all do.
My neuroanatomy is quite poor, right?
So this is olfactory transmucosal SARS-CoV-2 invasion as a port of central nervous system entry in individuals with COVID-19. First author, Jenny Meinhardt. The last author, Frank Heppner. And here amongst the authors is Christian Drosten.
And we're told deep in the footnotes that Jenny Meinhardt, Josephine Radke, and Karsten Dittmeyer contributed equally, whereas Helena Rodbrook and Frank Heppner jointly supervised the work. So,
from various,
and then the other authors are on there too.
Various sites throughout Germany. All right, so we all recognize, I think, that there are a variety of neurological symptoms associated with COVID-19, loss of smell, loss of taste, headache, fatigue, nausea, vomiting. Over 30% of individuals have those. Also, cerebrovascular disease, impaired consciousness. But as they say in this paper, there's really no good proof for what they call genuine viral SARS-CoV-2 infection of the central nervous system. And how do you do that? You need to get autopsy material. Really, there's really no other way to do it. You can look all you want at cell cultures and organoids, but it doesn't really tell you what's happening in people. We've talked about that before.
Unfortunately, Vincent, it's easy to get that material right now.
Yeah, sadly.
In a sense, yes, there's plenty of it, but as we'll discuss later in the paper, it's not necessarily something where when the pandemic started, they could get immediate access to this. So there is an issue with a lot of these bodies have been stored for a while before analysis.
Were you going to say something?
Well, you just have to be careful about the system when you're talking about organoids because they're only, second trimester. They're not an adult brain.
Yeah, they're not great to do
Well, you can't use them for this virus. They're not applicable.
Because they're not late enough.
Right, because they don't have all the cell types. They're not.
Yet, as you know, people do use them.
Well, yes, which makes you wonder what they're thinking. Did I just open up the freezer and find a plethora of them? Or did I really understand the biology of the adult brain versus the fetal brain? Because if you did, then you would have closed the freezer door.
Maybe you want to publish a paper and get another grant. Oh, no, I didn't say that.
Yeah, that's probably what they wanted to do. But as I just explained, I'm not an advocate of that.
Anyway, other coronas. Sorry, who are you, Alan? Sorry.
I'm Alan, yes.
Go ahead.
So in this paper, they went directly to the source. They went to autopsy brains, and they've got 31.
33?
33, and 31 of them were proven positive by PCR before death, and 2 were clinically consistent with COVID-19. And I'm assuming that was maybe due to a testing shortage that the remaining 2 weren't tested. But these are COVID-19 people who they're looking at autopsy.
And some of them had neurological alterations, right?
Yes.
Yeah.
of various sorts, like we already said. So
but they all died with COVID-19.
And so what they can do is a variety of techniques to look for evidence of virus. They can look by electron microscopy for particles. They can do PCR. They can look for viral proteins. They can stain the tissues. And in fact, you should take a look at this paper. I think it's probably open access.
It is.
Some lovely photographs of the various tissues that they're looking at.
Yes, they analyzed the cellular mucosal nervous micromilieu, which once you parse that, you realize what they're doing.
So it's very easy, as you'll see, it's very easy for them to find. So they look for the receptor. They find the receptor, and you know, this is with the caveats that we said before, but they do look for ACE2. They can find it in the olfactory mucosa, but no immunoreactivity in the parenchyma. That's the body of the CNS, right? Like the olfactory bulb and the medulla oblongata. No ACE2. So there's no way the virus is going to reproduce, right? Without ACE2. All right.
As far as we know.
As far as we know. So obviously they're focusing on the olfactory mucosa. There's a lovely picture of this in here because remember, in order to smell, you have nerve endings in your nose, and then they go through a piece of bone called the cribriform plate into the olfactory bulb, which has the neurons that can sense the smell or that can transmit the smell signals. And then from there, if a virus could get in, it could go elsewhere.
Right. And this is obviously, since it's in your head, a very close interface, a very short route to go from the air to the brain.
Yeah.
Right?
So if you look at the nice picture, if you picture the very top of your nose and then you go back a bit, where your brain is curving over, that's where the olfactory area is. And they look there and they say, this makes sense. It could get, because the virus is infecting the nasopharynx, right? So it makes sense that the virus could get in there. Of course, that has misled other investigators in the past. People used to think polio virus got into the brain that way, but it doesn't. What other viruses?
Well, some people might think that EV68 does that too.
Yeah, the virus Amy works on EV68. It's been suggested.
So the olfactory mucosa we're talking about essentially epithelial-like tissue lining this cavity. And I'm asking here.
Oh yeah. Oh sorry.
So it's, that I would expect virus to replicate there if it had access. And if we had, actually, this is the flip side to the ACE2 paper we just had. And that is that you do, in this particular case, where we have a very specific virus receptor interaction, if you don't have the receptor, you're not going to infect. Okay.
I think
it is a requirement. Okay.
Mm-hmm.
And then the olfactory bulb is nervous tissue. Is that correct?
It's on the other side of the bone of the skull, right?
Right.
It's inside the brain.
See, I don't understand the neuroanatomy at all. So do the nerves then project through into so that they're in proximity with the olfactory mucosa?
Yeah, the axons go through.
So that way, if I can somehow get to those nerves, that gives me a route to the brain.
That's right.
Right. So you have retrograde axonal transport. So you bind to the axons, the dendrites or the fingers, the spines of the axons.
And did we already say whether or not those nerves have ACE2?
They don't. They're not supposed to. They're not believed to have ACE2. Others have looked. It's been extensively looked at. So like Data at Harvard has looked, and various others have looked.
But here they say they find ACE2 in the olfactory mucosa, although we don't know what kind of cells.
We don't know what cells. But like Madeline Lancaster, who is the grandmother, or she's very young, but she's the mother of brain organoid fields. And so she's done a lot of these experiments in her Cell Reports paper. And she does not see any ACE2 either. So she correlates completely with Dada's work.
With no ACE2
on the tip, on the neurons.
On the neurons. But the mucosa, we're okay with that.
Yes.
Yes.
Yeah. Right. In fact, Data, who was on TWiV twin number 5 a while ago said he finds the receptor on the support cells, like the epithelial mucosa, but not on the neurons themselves, which is different from what they find here, right?
Right.
Here they find it on the neurons, and so I don't know why there's a difference.
I mean, I don't know.
So this tissue is not acceptable for electron microscopy, I take it, because
there are some electron micrographs.
There are some electron micrographs.
So there's no evidence for particles of virus
in one patient, they have particles, and these are some of the most convincing electron micrographs that I've seen. And I forget exactly what tissue we're looking at with the particles in it.
That is, there it is. Panel 3C is, it is olfactory mucosa.
Mucosa,
okay.
Right? So oral mucosa is susceptible to infection, right?
Yeah.
Well, so is
So
olfactory
I don't think there's.
also.
Excuse me.
So is the
I missed it.
olfactory
Amy?
milk also.
Yeah, I know. So they're all related to each other in terms of their epithelial cells, and they've got the same basic membranes, except that the olfactory epithelium probably has a different set as well to interface with the nervous tissue.
All right, so here's what they find. Looking by PCR, they can find viral RNA within the olfactory mucosa right below the cribriform plate, which is the bone part of the skull where the nerves go through. And not in many other places. They find some in cornea, conjunctiva, oral mucosa. Cerebellum is only positive in a few. That's the part back here, right? The part in the back.
And so it's 3 sections: the cerebrum, the cerebellum, and the medulla oblongata.
Right. So the nasal epithelium nerves go to the medulla oblongata. Is that correct?
Oh, they go all over the place. Fred Axel.
So they go to
Richard Axel actually did a lot of studies on that.
They go to the olfactory bulb, which is in the telencephalon region of the brain, which is in the front of the brain.
Got it. So none of the other tissues they looked at have, and in fact, the figure is really nice. Figure 1, we have a picture of a side view of the olfactory mucosa, the cribriform plate, the olfactory bulb. They even threw in the trigeminal ganglion there. It's for herpes virologists. And you see olfactory mucosa, and they have graphs of RNA, a lot of RNA in the olfactory mucosa. Then they look in the olfactory bulb. Some, not a lot, but there's some, much less than the mucosa.
And only in a few
patients.
So a lot of the patients, there's none in the olfactory bulb, and then a few patients, there's some.
And then,
so not everybody gets smell disorders during this infection.
Many people do, yeah.
Not everybody.
Not everybody. That's what I'm saying.
Right.
And then the olfactory tubercle, which is behind the bulb, is just one person had it. So it seems to me that it's clear that the olfactory mucosa is infected, but beyond it, there's not a lot.
Right.
But there's some. I think that's their point.
There is some.
There is some.
But if you kill off those cells at the level of the nasal mucosa, doesn't that stop the transmission to the nerve?
Which cells, Dixon?
The nasal epithelium. The nasal mucosa, the mucosal cells. If you kill those cells, then there's no way for that to.
I guess if you killed all of them.
Well, some of them, not all. You don't. I mean, there are thousands of different connectors in the nasal passage. Each one has a specific kind of receptor that tells you can smell benzene or alcohol or vanilla or stuff like this. So the ones that are killed off that are linked to taste, those are the ones that get killed off in only a few percentage of the patients. Let's say 20%, something like that.
Sure.
So the other mucosal cells might be killed off also, but it didn't affect taste and smell because it was the wrong set of epithelial cells. That's my interpretation.
Okay.
May I?
Yeah, it could be that, or it could be that you only need to kill off 20% of the cells, and then your ability to taste and smell goes down. So
yeah, there's no need though to go any further than that. Is what I'm saying. You don't have to go into the brain in order to stop transmission. You can stop it right at the interface
at the mucosa.
Yeah, exactly right.
Now they did see this interesting correlation where there's an inverse correlation between detectable SARS-CoV-2 RNA in the CNS and duration of the illness.
Yeah.
Yeah.
Which when I first read that, I thought, wait, so you get more virus in the CNS and you have a shorter illness. And then I remembered we're looking at autopsy brains. So it's a shorter illness. It took them less time to die, is what that's saying. So you get more RNA in the brain, you get more virus RNA in the brain when you've got a more severe infection.
So I'm looking at their last. They have a summary figure that summarizes the different assays that they use, whether there's PCR or immunohistochemical staining or EM or whatever, and they score whether each individual patient showed infection in the olfactory mucosa on the one hand or the central nervous system on the other hand. And it's apparent that what they've done here is to sort of pool and summarize the data. So you may have a few people who show infection in trigeminal ganglia, and a few people who, but say only 3 out of the whole bunch, and a few who show infection in the medulla oblongata. And there's 1, 2, 3, 4, 5 of those people, and those are 2 non-overlapping sets. Okay, but if you put them together, you got 8 people total. And so if you pool all the data where your conclusion is that there's some evidence of infection in the CNS, then what you come out with is that on the 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 out of the 33 are positive by PCR. Okay. So that's more than a few.
What do they mean by central nervous system?
Well, this is
the brain.
Yeah.
Not the spinal cord, but the brain.
But
any of these.
Is the olfactory mucosa part of the CNS? No. It is not.
No.
But the olfactory bulb is.
The olfactory bulb.
The olfactory bulb is. The trigeminal ganglia is. The medulla oblongata.
No, the brain. The central nervous system refers to the brain.
Yeah, they find it in those
These
places.
These are all parts of the.
But not in everyone, right? Not in everyone they find it in those, but mostly in the olfactory mucosa in many people, but again, not everyone, and then much less in these other parts. Okay, but their idea is that the virus is getting in through the olfactory mucosa because that's where they see it most, right? In those nerves, in those support cells.
Well, they don't even go as far as saying what support cells. That's data.
They just see mucosal cells, right?
They just say mucosal cells.
And they briefly discuss alternative pathways to the brain. For example, the blood. They say viremia is rare, and you have to deal with the blood-brain barrier as well. So they see this as a likely pathway to the brain.
But again, it's important to note these are people who died. So this is worst case, right? And looking at this, I would say this is not a neurotropic virus, right? I mean, we see in a subset of the people who died from it, there's detectable viral RNA in the brain. The receptor's not there. It goes to different parts of the central nervous system. There doesn't seem to be any consistent path that it's going from the nose into the brain.
I'm not sure that Ken Tyler would agree with that.
I think
he has a review describing a lot of patients, and his review focuses on various neurological complications.
Right.
So the loss of smell is one of them.
Sure.
But I believe he talks about a couple of cases that were encephalitic and maybe potential Guillain-Barré syndrome or some other things. And I think in one part he says that there has been viral RNA isolated from the CSF, which is, as an EV68 virologist, is the gold standard of what you need to define a neurotropic virus.
Okay, even if it's in a minority of cases?
What?
Even if it's in a minority of cases?
Well, it's like 4 out of the 650 cases of EV68 has RNA been found in the CSF.
Okay.
And less than 1% of children who are infected with EV68 form paralysis. And you should know, Alan, as coming from Vincent's lab.
Polio lab, yes.
How many people develop paralysis after a natural polio infection? Not very many.
Very small number, right.
Yeah, yes. So this is on par with that.
It's a facultatively neurotropic virus, yeah.
So we call polio neurotropic because it has the capacity.
Sure. Okay,
but, it's most of the time it's not going in the CNS, right?
Right. It's not, contrast that with herpes simplex. Yeah. Where going into a nerve is pretty much the plan. Yeah. It goes into the trigeminal or whatever, and that's part of its normal biology. So I would say, all right, amending that this may be a neurotropic virus, but I think probably in the majority of cases it doesn't go there.
Does the paper list cause of death for all these people?
COVID-19, I assume, but
no. What like heart failure?
Well, everybody dies of heart failure. It's
no. This is but the heart tissue is attacked by this virus, though. We know that because they've got ACE2 receptors in heart tissue. So it's more likely that the cause of death was not neurologic.
Right.
They don't actually.
That's what I'm driving at.
Some of these patients they don't know a lot about, unfortunately. But they don't mention that, Dixon.
So what do they put on the death certificate then? What does a physician put down on the death certificate?
They put down a sequence of things. So they would put down, a heart failure due
respiratory failure,
lung failure due to this, due to COVID-19.
Right.
So.
But isn't going into a coma not really part of this syndrome?
Going into a coma.
I don't think I've never heard that mentioned, at least.
No, right. So it's not from what I understand, it's not typically a neurological primary cause
so this paper is actually looking for why smell and taste get knocked out by the virus, perhaps?
It's looking for generally what is the virus doing in this interface in the nasal epithelium. And I suspect the knockout of smell and taste is what more or less a mechanism that Dixon was getting at, where it's killing off the mucosa that you need, the mucosal cells that you need to process smell. And if you lose smell, you're going to lose taste, because if you can't smell things, you can't really taste them.
Sure. It's not just smell and taste. There are a variety of neurological symptoms.
There are other,
go up. Okay. Confusion. They cite vomiting, for example, as a possible neurological dysfunction. And,
but don't we also have to deal with the cytokine storm events and their effects on those functions?
And that's a fundamental question here: are those apparent neurological phenomenon a secondary effect,
right?
Or is the virus getting into the central nervous system? And their answer here is, at least in some cases, we can detect virus in the CNS,
but it doesn't seem to be there reliably. Whereas these symptoms, so loss of smell seems to be not a universal symptom, but a very, very common one.
Yeah, but you don't always lose, you don't always lose the loss of smell in the same fashion. There are many pathways to making the chocolate chip cookie.
Right. Yes. Okay.
So after you recover, though, and your sense of smell has gone because of the acute infection, do you then regain your sense of smell and taste?
Can take a while.
Because there's some diseases that you don't.
Reports vary. So I've heard that there are cases where people haven't regained it yet, but remember we're less than a year into this pandemic, so people may have only just gotten over it last month. And I've heard directly from people who have regained their sense of smell and taste, but there are other effects, that may linger, and bizarre stuff. I would say, looking at this paper, that something common like loss of smell as a symptom for COVID-19 is probably not being driven primarily at the neuron level. Because even in the cases where people are dying of it, that doesn't seem to be a reliable thing that's happening.
Let me read the summary here. Our findings provide evidence that SARS-CoV-2 neuroinvasion can occur at the neural mucosal interface, may be followed by transport along the olfactory tract, thus explaining some of the well-documented neurological symptoms, including alterations of smell and taste. One caveat to note: the relatively long post-mortem interval an almost insurmountable obstacle in autopsy studies. And that, of course, complicates because things degrade, right?
Yes.
Yep.
I don't think they're ever saying that Dixon, I don't think they're ever saying that you die of neurological complications.
Yeah.
Right.
You die of the pneumonia, you die of the heart failure, you die of something else. But they wanted to know what, like, could you find virus there? Like, is it there? And that's, you can't just come in and say, I have a loss of smell and hack off your head and, then say, okay, well,
Here's why you lost your sense of smell.
That's right. You lost a lot of other things just now also.
Right. And unfortunately, these have not been modeled in animals.
Right.
That's the key. You need an animal model to study this, right? To see if neuroinvasion occurs, and if it does, where does the virus go and what are the consequences?
Yeah, and at the end of the discussion, they even call for more mechanistic studies to identify which of these mechanisms and some of it might be due to virus invasion directly to the CNS. Some of it might be due to other mechanisms that are going on in the course of the disease, and that needs to be worked out.
Amy, do any models show neurological disease? I think the original ACE2 transgenics.
Yeah, there's a Chinese ACE2 transgenic group that did a ACE2 transgenic. Barrick has a paper in Cell that talks about his K19 or K18 ACE2 human having, or no, that's Perlman. Maybe Barrick also has it. But Perlman's Nature paper recently that talked about the animal model says that you find virus in the brain.
Where, in what kind of cells?
I don't know that he dissected it down to that.
Oh, so he just ground up the brain and finds...
Okay. I think so.
Okay.
He might be doing it now. But, you had to overcome the barrier of the fact that there's a preconceived notion from the original SARS where you put the human ACE2 receptor in those mice, and then all of the mice died of lethal encephalitis, which you wanted, now you have to come back and make sure that that's not the same mistake you're making here.
Okay. Yeah. I think Barrick and Stanley both talked about that on TWiV. Yeah. Okay. Let's do some email. Amy, can you take Susan Weiss's email?
Sure.
Because I know you're good buddies.
I like Susan. She was I had a great conversation with her in Minnesota. I spent a lot of time with her at ASV. So Susan writes, I just listened to TWiV 688 and have some comments. PLLP or PLL Pro, trivial fact, MHV, which is mouse hepatitis virus, which is the coronavirus that Susan has spent many years understanding because it's very interesting. It's neurotropic. Actually has two PLPs, and as we call them. The NS3 protease and D-ubiquitinase, or DUB, are activities that can be separated, at least for MHV. And Susan Baker, who's at Loyola, who I know also, has had a paper on this last year, Dang et al. NJV. This is important because in the paper you talked about if they mutated the protease and the DUB activity, how can they separate the effects for poor replication and innate immunity from those of the DUB? I would think inactivating the protease completely would be lethal. More generally, there are so many conserved host antagonistic activities encoded in the ORF1A and ORF1B. For example, EXO-N, ENDO-U, NSP1, microdomain capping enzymes. Then there are accessory proteins that differ among the sublineages of the beta coronaviruses. SARS and SARS-2 are similar. MERS is completely different from it, as is MHV. These replicase proteins have sometimes overlapping activities and sometimes have additive activities with accessory proteins. For example, in MERS, ENDO-U reduces double-stranded RNA accumulation, and NS4B protects double-stranded RNA. In MHV, both ENDO-U and NS2 phosphodiesterase must be active to prevent RNase L activation. These viruses are very clever, and it is difficult to point to one activity as the major indicator of host antagonism. We have a paper in revision now where we found, maybe surprisingly, that SARS-CoV-2 activates RNase L and a PKR pathway and PKR pathways, while weakly including Interferon, in contrast, MERS completely shuts down all these pathways. We haven't worked on SARS-1 because it is a select agent. We do not have approval. RE, the RNA paper. If these mRNAs are indeed in lipid-bound vessels, they should also have genomic RNA in them. If you stain cells for genome, and mRNA genome is near ER and double membrane vesicles, but mRNA is cytoplasmic, presumably being translated. Well, you can always look for the ribosome. I would expect more genome than mRNAs. It is really too bad that they did not more broadly do some biological experiments. Remember that too in HMV, we see RNA genome and mRNA persists in the brain in non-infectious form. Nobody has figured out how this happens. Well, that would correlate with Diane Griffin's idea that RNA is persistent, and that's why it's not really a good marker for viral replication. All this gets back to the production of viral RNA from cytoplasmic sensors, a question far beyond coronaviruses. While viruses protect their RNA during replication, some of it is sensed by MDA5, PKI, OAS, etc. I don't remember if you've discussed Eric Schneider's paper showing that double membrane vesicles, she thinks it was done with MHV, have channels so that they are not completely closed and things move in and out, although they do not know yet. What yet? Keep up the great TWiV, Susan. So actually, we wrote a grant with Rod on the generation of double membrane vesicles.
We did?
The first reiteration of our SARS grant.
Okay.
And we talked about how that the collapsed convoluted tubules and the membranes being connected, and inside they're being protected RNA and stuff. So, and how when you adapted the virus to different hosts, you had all of these non-structural proteins that were forming these complexes with the alterations within them.
Okay. Rich, can you take the next one? Only because it's from Rich.
Rich writes, hi, TWiVers again. Follow up from my previously covered email. This article illustrates the travel of the virus and droplets around rooms, looking at 3 case studies. It mostly illustrates what I was getting at in my initial email to you, but I guess we now think that COVID-19 clouds or COVID-19 fog, I would say SARS-CoV-2 clouds or SARS-CoV-2 fog, might be likely. So amply fresh air purging the rooms will really help by diluting the droplets / virus in the air, reducing likely dose others could receive in any given amount of time. He gives a link to a news article, which we'll come back to. Thanks again for providing us with high specificity, very accurate and adjuvated informational booster shots twice a week. The side effect, grumpiness, wears off quickly. Efficacy is proving to be exceptionally good. Cheers, Rich, who adds at the end, still on our little island, Bali, where our very much outdoorsy lives and sea breeze might be helping us a bit. 29C and mostly sunny, though wet season is looming up early as it seems. I have not looked at this link he sent.
Yeah, you have to. So it's one of these where they formatted it so that you first you have to click to agree to eat a jar of cookies as you or something like that. So you accept cookies and then you can scroll down and it colors in who got infected in these various situations.
So people sitting at a desk, and 9 of 13 employees sitting at this desk were positive. Some of them were very far apart.
Well, it just trafficked. It doesn't
right,
say
so
that the person at the far
about,
left and infected the person at the far right, right?
Yeah,
he infects the person across him, and he infects the next.
And if you scroll down further and further, it shows how far this spread, and it spread a lot within that room. And then they give another example of a wedding banquet where the air conditioner was circulating the virus, and you see the people who were sitting in the flow of that air were the ones who got infected, whereas people who were sitting someplace else that wasn't directly in that airflow did not get infected. So it all supports the idea that you've got especially droplets, but possibly aerosols as well, traveling on these airflows. And it then documents or suggests ways that these could have been mitigated, separating people, having better ventilation, that sort of thing. So it kind of highlights how the epidemiology fits with the recommendations.
Yeah, and actually the graphics here are very good.
The graphics are quite good, yes.
It's really good.
They talk about virally charged microdroplets that patient O was expelling into the atmosphere. So, yes.
Didn't know I could do that.
Air conditioning, moving air around can propel, right? Because it takes the droplets and moves them very quickly.
Yeah, but they're saying you need to have better ventilation.
You need to have, so you need to have air circulation, but you need to have it not be blowing
directly
droplets from one person to the next, which is a place where masks could come into play. Although at the wedding banquet, obviously, you have to take those off to eat. But if you can stop the droplet spread over those short distances and those types of air currents, then overall increasing the air currents through the room or moving the whole thing outdoors is going to make a big difference.
So they have this bus where the recirculation of the air was on recirculation mode and people from front to back. So that brings up, in airplanes, they don't do that. They go top to bottom. And that's why the virus doesn't spread up and down the plane. Is that correct in my assumption or my understanding correct?
That's my understanding, except that about half the air is being recirculated, but it's being recirculated through a HEPA filter, as we were notified by an airline pilot who wrote in and talked about the air circulation system in the current Boeing and Airbus aircraft. In the planes I fly, there's basically a hole in the floor with a lever over it that you open it up, or there's a vent up here, so it's just right to the outside. But I'm the only one in the plane, or my family.
Have you flown lately?
I just last Saturday I went up, did a little bit of practice just to keep my hand in. The problem now is, it'd be nice to go somewhere, but there's no place to go because I can't go anywhere from Massachusetts except Hawaii, and the planes I fly can't get there. So.
Oh, don't focus on the details.
Yes, right.
Alan, can you take the next one?
Sure. So this is Bob. Hello, TWiV. Thank you for your amazing podcast. I've put many other podcasts on hold to give yours top priority. Near the beginning of TWiV 687 with guest Peter Hotez, he and your team acknowledged several motives for various groups to be involved in anti-vax campaigns. Conspicuously missing among them, or from Peter's recently released paper that was linked in the show notes, was actual vaccine injury, though you did touch on vaccine injury later in the show. The few people that I personally know that are anti-vaxxers claim to have experienced vaccine injury, including death of loved ones. They were not anti-vaxxers prior to the injury, or else they wouldn't have received the vaccine, but they are very strong voices and their stories get tremendous traction in the movement. In this kind of discussion, how can we especially make sure that those that are legitimately injured by vaccines are not thrown under the bus with all those sporting the nefarious motives you appropriately listed? TWiV has acknowledged vaccine injury in previous episodes, for example, the Cutter incident. I also recall Vincent stating in a previous podcast, I am not an anti-vaxxer, prior to listing some statistics for vaccine injury, such as the historical death rate due to smallpox. So let me also say that I am not an anti-vaxxer. This year, on turning 60, I pursued and received 3 different vaccines. A related question, could you discuss in some more detail the AstraZeneca cases that caused the 2 pauses to their trial? So far on TWiV, you had briefly acknowledged that those conducting the trial had concluded the incidents were not related to the vaccine. You've also acknowledged the truth that trial participants can coincidentally suffer from some disease unrelated to the vaccine they had received. But given Dr. Paul Offit's comments and gives a link, a deeper dive is warranted. Transverse myelitis, quoting from that site, and I guess quoting Paul Offit, transverse myelitis, which the study volunteers believed to have developed, typically occurs at a rate of 1 in 200,000 people, Offit said, so it would be unusual to see it in a trial of 9,000 individuals. Those odds above are less than the typical p-value used for significance, 0.05, but they are most especially so when the odds of appearing in a time frame close to the vaccine administration are taken into account. I have taken a crack at such a calculation that incorporates timing and would be very interested in yours. When the process of how vaccines cause injury is not well understood, theoretical discussion about how the injury could or couldn't happen should not trump statistically significant data. That is, an assumption that the event is related to the vaccine should be upheld. Besides the obvious prime compelling motive to prevent harm, safer vaccines are also a very important way to keep the most powerful stories, the true ones, from joining the ranks of the anti-vaxxers, which would cause yet more harm. I always look forward to your podcasts. Bob, who's in Drumbo, Ontario, Canada. And I think we don't have a lot more details on this AstraZeneca case, or really on the AstraZeneca trial in general. They've been kind of the most opaque of the 3 trials that have been announced so far. And this case, what we know is what we've been told, which is what's in the news, which is what AstraZeneca has said. So we're going on the assumption, which I'm not entirely comfortable with, that the people who are monitoring the trial got it right, and that they have good evidence that this was not vaccine linked. Now, I can certainly imagine ways that you would determine that apart from calculating the odds. So yes, in a trial of 9,000 people, maybe it would be very unusual and unexpected to see a condition like this. But if you then discovered that this patient was somehow predisposed to it for some other reason, then you might say, okay, in this case, this particular patient was kind of maybe expected to have this complication, and maybe we should have caught that in the initial screening. And so we've got an alternative explanation that doesn't involve the vaccine. Or maybe they were developing symptoms of it before they got the vaccine.
So I think that
those are hypotheticals.
But I think that one of the people who caused the AstraZeneca pause is who was quote unquote reported to having developed transverse myelitis, I believe was already on the pathway to developing multiple sclerosis. And transverse myelitis is a step in that pathway.
Right.
A very important, critical step in that pathway. So I think that they concluded that it was coincidental because they had this history of multiple sclerosis and in the process, and they were on the potential watch list for development of multiple sclerosis.
Got it. And that is somebody you would want to include in the trial. So I take back my statement about maybe we should have excluded them from the screening, because that's somebody you would want to vaccinate.
Yeah.
I think his other point about, I'm sorry, Rich.
No, go ahead.
Some people get vaccine injury. It's pretty rare, and we don't mean to exclude those. We don't certainly lump them with anti-vaxxers, but some of them don't want to be vaccinated anymore. However, I would say, if you have a bad experience with one vaccine, it doesn't mean that all the others are going to be a problem with you. So you need to talk to someone about that instead of saying, I'm not taking any more vaccines, right? So, if you got paralyzed from polio vaccine, that's horrible, but it doesn't mean you shouldn't take flu vaccines, right? Anyway.
And in fact, built into the questionnaire that you answer for a vaccine, like the flu vaccine is the one where this comes up, because people who are doing the right thing get it every year. And they ask, have you had a bad reaction to an influenza vaccine in the past? And if you answer yes to that, then you're probably not getting the shot, because that is a risk factor for maybe having a bad reaction in the future. And bad reaction does not mean, oh, my arm hurt a little bit for the day. It means like one of these things that went to the vaccine cord and you were seriously injured by the vaccine. As we've said repeatedly on TWiV in the past, it can happen. These are exceedingly rare events, and you're still better off getting the vaccine than the disease. But yeah, this is a really good point that we need to pay attention to these cases and note that these people can become extremely amplified voices in the anti-vaccine movement, because it is, that is one legitimate complaint.
I don't know if their voices. Oh, go ahead.
There's also a lot of, what happens very frequently, in particular in the anti-vax nation, is a lot of anecdotes of people who say, I got vaccinated and then I developed this or that. and they correlate that, and they make a story out of it. And as we've talked about many times before, you can get up in your white coat and spew data all you want, and people just go blank. But if you've got a compelling story, even if it's just one incident, and even if it's irrelevant, that gets more traction. And I want to remind people that there is a way of, in this country, tracking vaccine adverse effects that's routine. And that's the Vaccine Adverse Effects Reporting System, where anytime there is such an incident, it gets reported to a giant database. And so if something happens frequently, it doesn't, these are just anything that could possibly be associated. Not necessarily that
anything that correlated, that was linked in time. And
this is a two-edged sword, because on the one hand, its purpose is that if anything shows up frequently enough, it sets up a signal, and people look at that and they say, "Oh, we need to look into this in more detail." The downside is that if you are prejudiced against vaccines, you can go to the Vaccine Adverse Effects Reporting System and you can find any sort of adverse effect that you
millions of reports. Okay. Mr. Jones got his flu shot and was then hit by a bus. I mean, it's really
typically so-and-so got their human papillomavirus infection and then died. Okay. And there may be absolutely no relationship at all. So yes, we keep track of this. It's important to keep in mind that vaccine adverse effects, some serious, can happen, and we keep track of it. But it's important to look at the stories with, shall we say, an open mind at least, or a broad mind, and have a close look at these.
Dixon, can you take the next one, please?
I can, and I willingly do so. But first, I must inform you all that Marlene and I yesterday at a CVS attached to a grocery store got our flu shot.
Very good.
So it's about time, I would say. We've been putting it off for no particular reason up until now.
Did you get the high
you
dose?
Probably got the old
guys. So far, no adverse responses, except I'm a little bit slower in dismuting. Niall writes, an application for Rich's Chernobyl fungi. Hello again. I thought you might get a kick out of this. I found it quite exciting. Humans on the moon and Mars would face the problem of damaging space radiation, but new research suggests one possible solution to the fact that space wants to kill you, according to CNET. To protect astronauts, scientists have been studying an unusually hardy organism discovered in one of the most radioactive places on the planet, Chernobyl. In some parts of the plant, the level of radiation spiked so high that exposure would kill a human in about 60 seconds. But several species of fungi have been discovered in the reactor, and they're thriving, feeding, in quotes, on the extreme levels of radiation. A new study, yet to undergo peer review, was published on the preprint repository BioRix on July 17th and examines one of these species, Cladosporium spherospermum. It suggests the fungi could be used as self-healing, self-replicating shield to protect astronauts in deep space. Researchers placed the fungi aboard the International Space Station for 30 days and analyzed its ability to block radiation. The proof of concept study showed that the fungi were able to adapt to microgravity and thrive on radiation, decreasing the levels by almost 2%. One of the major advantages, the researchers write, is the fungi self-replicates from microscopic amounts. You would only need to send a small amount to orbit, give it some nutrients, whatever that is, and let it replicate, forming a biological radiation shield. With some tweaking, the fungi could be used to shield bases on the moon or Mars. It's a long while until we put boots on the red planet, but the groundwork is being laid now. Hope you enjoy. Niall.
Nice.
So
cool.
What are they going to do? Coat the The buildings with this stuff?
Coat the building in mushrooms. It'll be like the 60s. Man, just camp out under the mushrooms.
And they glow at night when you turn out the lights. Right.
Let me see.
Maybe, I don't, 2% is not a lot, right?
No.
So I don't think the fungi prevent radiation from going through them. What this says is the fungi are not affected by the radiation that goes through them.
Well, I think the 2% reduction, they are shielding some of it.
A little bit, but really, that's 98%. I think for humans, that's not enough.
You know what you would need, Dixon? You'd need a vertical farm of these fungi.
You would. You absolutely
the layers would prevent it.
Well, you know what? That's possible, but that's a lot of nutrients you're asking for here.
I have a friend who just retired from his, younger than me, so retired, left his faculty position to go work at the NASA Ames Research Center in the synthetic biology division, where their goal is to produce critical products for deep space missions to Mars.
Right.
Okay.
Nice. Is that Ames? Is that Ames, California?
Ames, yes, it's in California.
I've met that guy. Pete Warden actually runs the whole facility. It's associated with the Singularity University. It's a wonderful place, actually. Great stuff.
So these critical things include potatoes?
So engineering organisms. This is synthetic biology, right? Engineering organisms to produce critical products. I don't know. Any kind of carbohydrate would work for me.
You going to Mars? No. You don't want to go to Mars?
No, you're gonna need some protein too.
Yeah, true.
We asked Kate Rubins the first time she was on TWiV, would you go to Mars? And her answer was, Rich,
depends on who I'm going with.
It's really funny.
It's a good answer. That's
a good answer. She had good answers to a lot of stuff. That was great.
But this is what the Ventnor Institute does, or at least part of it does.
Part of it does synthetic
biology, like how to find new fossil fuels.
Cool.
So, when I was in Chicago, I heard this talk about this guy who was manipulating E. coli to eat some kind of garbage that, like, I guess the plastic in the garbage or something or other. It was really it was really cool and interesting, but it was like, I don't know how this is all going to work out.
Yeah, there are a lot of these very, very challenging, like material life cycle problems that you confront if you're going to actually go out of Earth orbit and spend any significant time. I mean, the current method for astronauts to do their laundry is to pack up the used underwear and such and shoot it into the atmosphere to incinerate.
Fantastic.
And they just put on fresh underwear. So
that's not being done anymore, though, Alan.
It's just all disposable.
That's right. They changed that modus operandus when they became publicly known. Look up. Oh, there goes the international. What's that streaky stuff coming out of the back?
No, that's just it incinerates. It burns.
That's done.
Well, Kate is listening.
Yes.
Okay. So, Kate, how do you do your laundry?
How do you do your laundry now? Have you updated that?
It's a takeout. Quite literally.
All right.
But yeah, if you're going to Mars, you've got to recycle everything. So you're going to need genetically engineered organisms
Obviously,
that can handle all that.
I can't control these people.
Really? No. Whatever made you think you could?
No, I don't
I'll
want
tell
to.
you what. If I'm going to Mars, I'm not wearing any clothes.
Let me know where you're going. I might be looking the other way.
You're going to moon on the Mars?
Why don't you want to wear clothes to Mars?
Just learn how to dispose.
That's right. That's right.
Well, they're going to make bacteria that are embedded in your clothes, and they'll automatically clean them.
Well,
that would be good.
Nice. I wrote her after we talked to her because we talked to her about whether you could use human waste to feed the plants.
Right.
And at the same time, the plants could recycle waste, sort of an ecosystem. And she said, well, we're worried about, sterilizing the human waste. Right. We can't be scattering that around the space station.
Yeah.
Scattering.
Right. Good. So I wrote her back and I said, I wonder if you couldn't somehow expose the solid human waste to space for a period of time.
Irradiate it.
Yeah,
Yeah.
not only that, but extremes of heat and cold, the vacuum, and I wonder if that wouldn't sterilize it. Then haul it back in and feed it to the plants.
Yeah, but they have to recover the water from that.
It would
They've got
lose
to
a lot of water in the process.
That's right. You have to recover the water from almost everything. You notice that in The Martian, the movie, those were all lyophilized samples that he had to rehydrate in order to get them to work, right? So,
Rich, let me get this straight. Are you going to be the guy that does the spacewalk and brings the poop outside?
Hell yeah, I'll do that.
What do they do with it? What do they do with it now? Do they pack it up and bring it?
She told they actually
they dewater it first.
No, that gets incinerated. She didn't talk about the water, but basically that gets packaged up and shot back into the atmosphere and burns up.
Yeah,
that was on our interview with her. I think that's correct.
Yeah, they can't waste a drop of water.
Standard waste disposal approach for astronauts in orbit is to shoot things into the atmosphere and it burns up.
Right.
That's also the way we get rid of satellites at the end of their lifespan.
Indeed.
Okay. All right. Abby writes, just listen to the last episode. As a practicing urgent care pediatrician, the need for a central message with good information is so evident. People have no idea, with the first letters capitalized in each of those words. Just in the last week, I had patients who were sent to school with cough and congestion, developed fever at school. Another mom asked for the curative med to treat COVID for her kid. Her internist had prescribed a Z-Pack for the mom and said it will cure her. Another dad said his son had severe headache and fatigue and wondered if there's any illness going around. My whole family still has elevated resting heart rates. We aren't all better, and we had this in March, and most doctors are still gaslighting people like us. We are in trouble, pretending we can control this without more national education at every level. I'm trying a few patients at a time, and no, I did not prescribe Zithromax. Thanks for all the updates. Abby Siegel is an MD. Well, thank you, because we have no idea what's going on, really. And it is...
and thank you for all your work.
We have no idea. We do have no idea.
I get some direct reports of this because my wife is working in a hospital in Connecticut. And by the way, she's an exemption. She's allowed to travel to Connecticut because she's an essential worker. But the exemption is different in the two directions. Because when she comes home to Massachusetts, she's coming home. So she's not an essential worker here, but she's allowed to be here because she's commuting weekly to another state. It's insane. But yeah, they're COVID testing everybody and seeing the rates go up. And they've had issues where since she's in psych, they're trying to manage admitting people to the psych hospital because remember, ordinary diseases haven't gone away just because there's COVID-19. So people are showing up with serious mental illness issues. They need to be in a psych hospital. And if they also have COVID-19, what do you do? And the answers to that question are not reassuring because there's just not a lot of good infrastructure to handle this sort of thing. And it's going to get worse.
Well, I think Daniel has, at least in conversations I've had with Daniel, people refuse to get tested.
Yeah.
So you send your kid to school and the kid says, I have the kid has sniffles. I mean, ignorance, you claim ignorance. You say, I
didn't know. Right.
Yeah, exactly. And yeah, you're telling the truth. You didn't know, but it's kind of, I didn't know with an irresponsibility parentheses next to it. Or whatever. Well,
and because of the lack of a coherent unemployment policy and such and assistance to handle this, a lot of people are now in a situation where they have to send their kids to school so that they can go to work. And that's making things worse.
Well, yeah, that's what Daniel's complaining about, is they're specifically not getting tested. They're offered a test. They say, no, thank you. They walk away because they know
they would rather not know.
Right, because they won't be able to go to work, whatever that entails.
Plausible deniability.
Right.
Yeah.
And I believe that I read an article earlier in the pandemic in your neighborhood of the woods, Alan, where a mom specifically sent her kid knowing that the kid was COVID positive, was SARS-CoV-2 positive.
Okay.
And I can't necessarily, I mean, as I say, a lot of these people are backed against a wall, and I can't necessarily say that they shouldn't do that, because what were they supposed to do?
Which is, I think, part of what I was complaining about or pointing out when we talked about de Blasio's pandemic center, was that you have to have this social economic consequences built into the plan. It should be 3% about the science and 97% about the people, not the other way around.
Yeah.
Let's do some picks. Dixon, what do you have for us?
Well, I have the latest edition of the National Geographic our best animal photographs, or images, I should say, because they're probably all digital, from 2020, and some remarkable moments in nature that have been, these people that take these pictures must have infinite patience, and they're just amazing. They're just getting more amazing every year. The photographs just get more personal. You can almost tell what the animal is thinking by looking at some of these pictures, and they're quite beautiful.
These are great, Dixon. There's the parasitic wasp.
Yeah.
Larvae on a caterpillar. Man,
That's it. And the feeding bees, all of the bees at the little line. I'm just fascinated by it, and I'm never bored with unique observations of wildlife, and I'm never bored with that. It's just absolutely fascinating. So that's
cool.
what I have.
Thank you. Amy, what do you have for us?
So I have a book. It's called American Contagions, Epidemics and the Law from Smallpox to COVID-19. It talks about how we change the law to adapt to our public health needs in an emergency. And this is actually very controversial at this particular point in time under this pandemic. Not clear, because all the other pandemics that I've lived through, from the smallpox pandemic of the late 1800s and the flu pandemic of 1918, this was not a question whatsoever. But you change the law a bit, or the practice or the interpretation of the law a bit for public health, and then you go back. And it appears that several of the very conservative, or not even very conservative, or originalists of the Supreme Court, like Alito, feel that this has been usurped and that we're not going to go back. And this is why recently the Supreme Court said that in New York, you could not have a restraint of how many people were in a religious service. It was a violation of freedom of religion, which, public health should usurp, and others have written in the dissenting opinions that public health should be put to the forefront in an emergency, and then you return, which we have always done. So it demonstrates that we actually don't know anything about history, and we keep repeating the same problems over and over and over again. And we should learn that you change a bit for dealing with the pandemic, and then you return when normalcy is restored, or whatever you want to define as normalcy. So when Vincent and I were discussing this, of course, the vaccine came into play. And it turns out that there was a 1905 decision by the Supreme Court called Jacobson versus Massachusetts. And Jacobson didn't want to be vaccinated because the vaccine had more severe side effects than our vaccines do today. And the Supreme Court intervened and said that for public health well-being, the law in Massachusetts usurps your individual rights. And so I don't know why we're still having this discussion today. So there's lots of things about this book that I thought were interesting. And I think really learning the lessons of history and reapplying the rules or stuff we have forgotten, and that this is detrimental, and we should all really take a step back and learn about what we've done in the past and how we've overcome pandemics. Because as I said, the pandemics that I've lived through, we've all survived.
So you're saying that the Supreme Court doesn't know about this 1905 decision?
I'm not saying that they don't know about it. I don't know if they know about it. I'm just saying it's precedented already.
Right. So lawyers can argue around precedents depending on their personal agendas. And For many years, people on the right used to accuse people on the left of judicial activism, which was, somehow departing from the law in order to advance a political agenda. But if you've read Supreme Court decisions from the Republican-dominated Supreme Court era, such as Bush v. Gore or Citizens United, you know that that's a load of horse manure, that the folks on the right are anxiously engaged in judicial activism of their own and advancing political agendas. And this recent Supreme Court decision is a perfect example of that kind of thing.
But we also, there is precedent. And
yes.
So the idea that we don't go back to normal is not correct. And that's what drives this idea, is that we're going to be, our freedoms are going to be taken away from us. And, oh my God, they're never going to be restored. Well, they've been restored in the past, if you know anything about history. And so this whole idea is wrong. And so maybe if we took history classes and we actually learned the lessons that we're supposed to, then we wouldn't be wasting our time having the same arguments, as I said, after my first pandemic, which was in 1890.
Dixon, you were going to say something?
Yeah, I was just going to add that, yelling fire in a crowded theater is not prohibiting that, is not interfering with personal freedoms and rights.
Right. No, our rights can be circumscribed for the benefit of society.
You don't have the right to endanger the life of another person. Right. And that's where the law should start, and that's where it should finish. But I would tell you that enforcing this law today in places like Williamsburg and places like the Midwest, where you have these large evangelical meetings and large weddings and large, even in the Native American communities out west, when someone dies, everybody from the community shows up to mourn, and that's tradition, and you can't interrupt that, even if you wanted to. It's a very difficult situation now.
But I believe one of the Native American communities is where the lowest rate of infection and deaths from from SARS-CoV-2 or COVID-19 are. I believe it's been documented.
Well, the Navajo Nation is not one of those places because they're really being hammered.
Yeah, no, but I believe that Saul sent you an article about this, about how it was being handled.
I just want to point out that with respect to this religious overturning of the Supreme Court, the Pope wrote, public health should predominate, not religion.
Absolutely.
Yeah.
Absolutely.
Alan, what do you have for us?
This has been in my list of picks for a long time, but just came to my attention again recently because they're doing another round of cool experiments. This is HamSci, which is an organization dedicated to citizen science using radios. So it's ham radio operators. I know we have a number of them listening to the show, but also, even if you don't have a ham radio license, if you're just interested in these earth science subjects like sunspots and the upper atmosphere, the ionosphere, there are these really cool phenomena that happen on massive scales that we have virtually no understanding of. So there are layers of the atmosphere above the part that we breathe, way, way, way up, that are full of charged ions and are critical for protecting us from radiation from the sun. We can actually study them by sending radio waves through them, and it turns out there's this whole group of millions of hobbyists who do this routinely called ham radio operators, and that's how we talk to each other around the world. We actually refract our signals through the ionosphere. Well, now that we've hooked all our ham radios up to our computers, it's possible to do these massively networked studies where you look at changes in the ionosphere in real time. And NASA does not have this kind of instrumentation because it has to be all over the place in order to do these sorts of studies. So they can do it from a couple of places and other organizations can do it from a couple of other places. But the hams who are sitting all over the place can probe it in ways that the professionals can't get to. So this is a setup for professional atmospheric researchers and solar scientists to collaborate with ham radio operators and shortwave listeners to do these types of experiments. They're doing one right now for the December 2020 eclipse, which you may not have heard of because it's not going to be visible in North America, but it is going to be visible in South America. They did one for the 2017 eclipse that actually documented dramatic changes in the ionospheric, propagation just in the brief interval as the sun's shadow was passing. And they've got another that's looking at a particular type of propagation that's slightly lower in the ionosphere that changes in the wintertime due to these very poorly understood clouds of ions that move around throughout the seasons. So this is something for people who want to do science projects that you don't even have to leave the house for. So even during the pandemic, you can participate in something like this.
Pretty cool.
It's a good project for students too, right?
Yeah, and they've had undergraduates and graduate students who've ended up getting, presentations at conferences where they took the data from this ham-sci type of network and managed to discover new things about the ionosphere and about solar flares and that kind of thing.
You got your ham license yet, Amy?
No, I haven't finished the paperwork. I'm busy plaque assaying.
Oh. Rich, what do you have for us?
I have a Nature News article called "It Will Change Everything. DeepMind's AI Makes Gigantic Leap in Solving Protein Structures." This has been in the news lately.
Yeah.
And I thought I would look into it and figure this out. So a significant problem in biology is being able to predict protein structure from primary amino acid sequence. It would be enormously useful because we got all these data around, with gene sequences. We don't know what they do. And part of the way to figure out what they do is to figure out what the proteins look like. From structure, you may be able to predict function, et cetera. Right now, the experimentation to determine structure is difficult. Okay, so it'd be nice to, God can do it, can make a structure from primary amino acid sequence. So maybe we ought to be able to do this from theory. So this is an approach that uses artificial intelligence. The artificial intelligence resource they use is called DeepMind, which is an organization that originated in the UK in 2010 and was bought by Google in 2014. So now it gets sort of called both DeepMind and Google, or Google DeepMind, or whatever. But basically, it's a mashup of the two. And there's a periodic event that's a kind of a meeting that's called the Critical Assessment of Structure Prediction, where various scientists are challenged to predict the structure of a number of different proteins for which the structures have been solved but not published. And then they assess how good they did. And apparently this new algorithm of DeepMind called AlphaFold 2 blew all their minds at the most recent meeting and got a score of near 90%. And they said a score of above 90 is considered roughly equivalent to the experimentally determined structure. So it looks like we're getting close to a breakthrough here where we're going to be able to use artificial intelligence to predict protein structure, which would be amazing. Yes. I think it's amazing in two ways. One, being able to do it, and two, the use of artificial intelligence to do this fascinates me.
Which, does it say how long it took them to do it?
I think somewhere in this it does. It's days to weeks. It's a long time. The thing I love about this artificial intelligence thing is, as I understand it, the most recent approaches to artificial intelligence are not that you give the computer a bunch of different little pieces to use to put it together, a bunch of little parts of a solution and have it put it together. You just give it the problem. Okay. And the computers are so sophisticated now that they figure out their own solution.
And it's okay for something like this to take a few days, because if you get it up and running, you could just have it grind away through the databases, prioritize your proteins and have it spit out a new structure. Oh, you were looking for this? You want to know what this looks like? And it would be wonderful to have something like that functioning. In fact, this just came up on the show today where we're talking about people analyzing sequence, and it would have been nice if they'd looked at three-dimensional structure of the ACE2 receptor and the spike protein. And so this is the kind of thing you could do with that.
Yeah, I was going to say, let them go at the ACE2 spike interaction, All right. My pick is a book about ants. It's called Army Ants, Nature's Ultimate Social Hunters by Daniel Cronauer. And Daniel was just on TWiV with Nels and myself, and that episode hasn't launched. Maybe it will by the time this episode does. But he's written this book. He's an ant researcher at Rockefeller. He works on raider ants, which are related to army ants, but you can't work on army ants in the lab. You have to observe them in the field. And they're amazing. Some of their hives are millions of ants. They go out and hunt other ants, insects and so forth. They are incredibly social. And there's one cool behavior. And he photographs himself. He's got really good photos in the book. He takes on field studies. The ants can make a bridge for other ants to walk over. So they'll all link together and span like a couple of inches, and then the whole whatever is left is walking over them, and they have no problem with that. They're okay to do it. And then when they're done, they get up and walk away.
They're probably griping the whole time.
And the cool thing about ants, of course, is that they're blind, but they do everything by smell. And so one of the things that Daniel, he's got a whole chapter in this. There are parasites that invade the hives and they live. They're insects and snakes and worms. They figure out how to trick the ants into not smelling them, and they can live there. And he said sometimes when the hives move, they'll all go somewhere else, and you can see these stragglers among them, that snakes and cockroaches and beetles, they don't belong, but the ants don't know they're there because they've tricked them. He calls them social parasites. It's very cool.
They're like the imposters in Among Us.
Exactly. I just saw a little segment on a nature show where a very common butterfly, the caterpillar of which is actually captured by ants and taken back to the hive because they're emitting pheromones that are exactly the same as the ants. And they keep feeding these larvae until they get gigantic. They look like queen ants. And then they hatch, of course, and fly away. And the ants are, what the hell was that? By the way, cool. Vincent, when I first read this very fast, your pick of the week looks like Amy ants.
Amy ants, that's right.
I said, Amy's a guest. She has ants. Isn't this wonderful?
Anyway, it's very cool. And his lab was the first to do a gene knockout in these raiders. They're clonal, so you can actually grow them in the lab. And they knocked out a gene called ORC. Is it ORCA or ORCA? And it knocks out their smell, and they can't interact with each other anymore. And there's one assay they do. It's called the Sharpie assay. They draw a line on a filter paper with a Sharpie, and if ants will avoid it, they don't like the smell. But if you knock out ORCA, they just walk right across it. It's really cool. So we called that episode Raiders of the Lost ORCA, because they're raider ants and the gene is ORCA.
I approve.
And I made that up.
Very good.
Unbelievable. I'm getting a little influence from Alan.
Alan approves that message.
All right, that is TWiV 690. We will have 700. The last episode of this year is going to be 700. How about that?
Wow.
Amazing.
We just had 600.
And the special occasion for 700 is we had a pandemic.
It is, and it's a viral pandemic, so
yes,
all really, well, it's very sad, but.
Yes, actually.
690, show notes, microbe.tv/TWiV. Send us questions and comments, twiv@microbe.tv. And if you'd like to support us, microbe.tv/contribute. Our guest, actually, she's a rotating host, right?
Yes.
Yes.
She is.
Amy Rosenfeld.
Amy, turn around a little bit and show them how you rotate.
Should I pirouette? Next time I'll bring my tutu and ballet shirt and toe shoes.
We'll see you next year, right?
You would love to see that.
Yes.
Amy's at entrovirus.net and Amy Virologist on Twitter. Thank you, Amy.
My pleasure. It's always fun.
Dixon Despommier is at Racaniello.org and thelivingriver.com. Thank you, Dixon.
Good.
It's fewer words today from Dixon. Right?
Yes.
That's right. I'm muted. Self-muting.
Rich Condit's an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Fair enough. Always a good time.
And Alan Dove is at alandove.com. On Twitter, he is Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.